opening speaker, the feature speaker, he mentioned neonics. What comes to mind to me with neonics is the phrase that we used to use was, it's a no-brainer. And it was also really easy. And everybody did it. And then PMRA caught on and went, there's too much of this stuff around. So it was not a no-brainer. There were fields you didn't need it in, but it was easy. This is not easy. You have to figure out, where does this belong? So our agenda quickly, an introduction. Obviously I'm not an IT guy. Then we're going to talk about who cares about biologicals besides you people. Different types of biologicals, what kind of testing goes into biologicals, how do different biologicals work, and then some grower experiences at the end, and then just a little bit about the future. Introduction about me. I'm the technical lead biologicals. That became a word for Syngenta 6 months ago. This is me, 1980. I go to the University of Guelph. There's a Midwest crop tour for a credit between your third and fourth year. The turnaround point is Denver, Colorado. So this is just outside of Boulder, Colorado. Rented a motorcycle, drove around through the mountains. But the purpose of this Midwest tour is you see all sorts of different farms. And it makes you wonder, why am I doing what I'm doing in Canada? Like, they have cobs of corn that are as long as your forearm, full to the end. And it's like, that's huge compared to Ontario. And it was an interesting thing. You're also sitting in the middle of a hot field, 35 degrees, and somebody's talking about breeding reed canary grass. And you're like, that's a weed where I come from. So this is how I got into agriculture. Last two people to farm in my family were my grandfathers. Anyway, I didn't have a family farm to go to. 1981, this is now, you remember what interest rates were in 1981? 19%, 21%. It was horrible. Job market, horrible. A lot of colleagues of mine, students, went to family farms. I didn't have one, went and found one in New Brunswick, milked cows for 3 years. It's not a lot of fun milking somebody else's cows. Ended up getting a job with Syngenta. 2 years later, I marry this young lady, she's from Nova Scotia. I work in New Brunswick in the Maritimes for 2 years. Then I get transferred to Ontario. She comes with me, thank you. And we've been married for 34 years with no time off for good behavior. Here, I'm in R&D for the next 20 years. I'm looking down at the ground. It's mostly herbicides, some fungicides. 20 years of being in R&D. And I thought that was hard work. And then my career took a 90-degree turn, and they said, we want you to go and be the technical person for the sales group. That was hard work, because now you're getting our salespeople technically knowledge. Like, they have to know what they're talking about. They have to be able to get more than a one-word yes or no answer. They have to be able to ask questions back to make sure they understand why you want to know something. So I had to hire 4 contract people. They're the 4 people on the left. 2 of them are still with Syngenta. One's got 15 years and one's got 18 years with the company. So I'm pretty proud of those guys. Used to like to travel when you could travel. This is the highway in Spain between Barcelona and Madrid. And that arch there, that is the indicator of the prime meridian. That is 0. So on one side you're in the east, and the other side you're in the west. I thought that was pretty cool. Then, after being in Ontario for 30 years, my wife retires from being a pharmaceutical rep, and it's just fair that we move back home to Nova Scotia, her home province, and we're now living in the Annapolis Valley. So that's me. The perception of biologicals around the world. In the EU, they are the leading consumers of biologicals. They use a lot of them. South America is the fastest growing area for biologicals. There's significant growth in Asia. Africa has 50% of the North America market, and North America is the largest market. So the future is coming. You are going to be able to use more biologicals, and they're going to be better. Right now, they're kind of so-so. We're just figuring out where they really work for farmers. Factors that influence the increase in biological use. McCain's, huge global company. They want to be seen as the good guys. So they want to have all their customers going, yeah, I like the way McCain's does things. People who grow for McCain's are trying to save the planet. Then you've got the government. That always helps. And they come in and say, yep, nitrous oxide, bad for the environment. We need to cut down on this. So we want people to start using less fertilizer. Then farmers turn around and say, fine, we can get some positive publicity out of this. We are actually working towards what you want. We are looking at microbes to nourish crops as climate pressure grows. And then the government kind of actually widens the scope and says, we're going to look at pesticides too. So if you're using a fungicide now or an insecticide, maybe even a herbicide, there might be a shift towards biologicals. So what do farmers think about these technologies, and in particular about the nitrogen fixation technologies? Stratus, I don't know if you've heard about them. They're an ag research company. They're the ones that say if you want a $50 gift card, answer these next 50 questions, so you can get a $50 card in half an hour. Answers they got back were 29% of farmers in Canada were going to cut their nitrogen rates in 2022. Either they couldn't find it, or they thought it was getting a little too pricey. 75% said, I'd be willing to try something that was a biological for nitrogen fixation. 39% that they thought the new regulations, when they eventually materialize, are going to be painful, so they should start figuring out how to work around it. 7% said, I'm actually going to try one this year. And we were lucky enough to have some of those growers use some of our products just to see how they were going to work. And then we were wondering, well, what's it going to take? What kind of difference does it have to make? A 5% increase in yield would be enough for most growers to go, okay, I'm in. So there are a lot of biologicals in the world, a lot of companies making them. And you know, a lot of these you might recognize a name here and there. You might go, I don't know any of those people. Valagro, it's an Italian company. They've been around for just over 40 years. Syngenta bought them because Syngenta said, we need to get into this market. We bought a market survey and it said there are 5 companies that are really good at doing this, and Valagro is either number 1 or number 2 on any given year. So that's in Italy. So we buy them. That's all good. We've got some new products we want to look at, make sure they work here in Canada. Azotic, they have a nitrogen fixing bacteria. We didn't buy them. We just came into an agreement with them and said, let us be your sole provider. So that agreement was signed early this spring, and it was really hard to get things going, but we eventually did to get some trialing going on. Acadia Plant Health, that's an outfit based in Nova Scotia. They basically go across the country with their product. Ours are slightly different than theirs. You can see Symborg just got circled over there on the far left-hand side. That's a German company. They have a nitrogen fixing bacteria. Corteva just bought them. So the big ones are figuring out this is the way things are going. And just like the feature speaker said, companies are watching, they're waiting, and then they're going, we either get an exclusive or we buy them. And then there's Pivot Bio. They're in the US. They have a seed treatment that's coming, which is for nitrogen fixation. And they're just trying to hire people in Canada to get their company going. There's probably lots more, but I didn't have time to research who's looking at who. So biological categories, there's 3 basic categories. Biocontrol, you might see the word biopesticide. It's the same thing. These are for controlling the usual suspects, insects, diseases, weeds. But it's going to be some kind of biological that does it. They may not be as effective, which means you might have to go, okay, I get 50% control. That's not very good. But if I do it again, now I'm up to 75% control. And if I do it again, now I'm up to 95% control, which is what I used to get with my old chemistry. So this might be the way of the future, that you're going to have to use these products more often because they're not as effective. Biostimulants. Oh, yeah, I forgot to mention. You want to register a biocontrol, you go through PMRA. Syngenta is really good at this. We've been doing it for years. We're pretty comfortable doing it. Biostimulants, this is about managing stress in your plants, not yourself. It's for heat, it's for cold, it's for flooding, it's for physical damage like hail. Biostimulants are a little in the gray zone. It's pretty hard to evaluate, how do I think my plant is doing today based on yesterday's weather and what I put on it to help it. That's a really uncomfortable zone for me because I was used to rating herbicides. This is working, that's not. This is working. This is totally different. You pretty much have to go through the whole season and go, how did it end up? And then work backwards and see what did I try and mitigate as a stress in my plant. Then there's biofertility. This is probably the newest area, except for the fact you've been doing it for a long time. Inoculants. They fit into this section. Everybody uses inoculants on their legumes. They colonize the plant. They make nodules on the roots. Now your plants get to fix nitrogen from the atmosphere. Endophytes are different. They don't just work on legumes. They work on all sorts of other crops. Now you have to go through the Canadian Food Inspection Agency for this. You'd think that would be easier. It is not. It's just as hard as PMRA. And that's basically the story in a nutshell. There are no shortcuts. Everything, just because it's a biological, you're not getting played favorites. It's all really hard to deal going through the mechanism to make sure it's safe for Canadians. So Syngenta has biologicals. They all showed up in the last 5 years. There's a whole bunch where a lot of people use them the most in the EU. Most of them are for stress. Some of them are for insects. And some of them are for irrigation water management. We have a couple of fungicides in South America. We have another fungicide in Asia. And we have a handful of products, or 4, in North America. And these are the bigger ones. Aflagard is for corn in the US. It's to stop aflatoxin from getting into their crops, especially in the south. Then we have Amatis, which is for root growth. It would have to go into the furrow if you're doing field crop like canola. Megafol is a stress product, and then Invita. I'm going to talk about Megafol first and then Invita. I'm talking about Syngenta products because those are the ones I know. You could probably say, I heard this guy talk about this kind of product. Talk to your company that you like dealing with and just say, do you have something like that? So, abiotic stresses. These are the records, and you can't go, well, the records can't all be 100. No, some person got 600 and some odd 100 bushels per acre of corn. Some wheat grower got 278. Some soybean grower got 175. It's like, wow, those are the records. Everything had to go right, and they were out there spoon feeding these things. What do we get on average? 24, 13, 22 of the record that we've ever had. The other 10, 5, 9, 9, those are biotic losses. Those are weeds, diseases, insects. You go out and you deal with those. The other ones, the big gold bar, yellow bar, that's the weather. That's where you don't have much control. And this is where abiotic stresses typically come from. And we're trying to get you to play in those big number areas and say, how much of that weather effect can I undo? Which brings us to Megafol. Less stress, more growth. This is Villagro's #1 product. We bought Villagro. It's been around for over 30 years. This is the 3rd version of Megafol that there is. It's not a 30-year-old product. It's something new all the time. And how they get it is they go to places where plants grow in harsh environments. First time I saw this, I thought, you had to say seaweed. And I'm thinking, ugh. And then they explain why. They say, okay, here's a plant. For 6 hours, it's in cold, salt water. Then for the next 6 hours, it's in the air, and it's exposed to ultraviolet radiation. Then it's back to the water, back to the air. A plant that can survive that must have some pretty cool stuff that it makes to be able to live there. Picture a canola plant and do the same thing. I figure half a turn and you're done. So something that can live there probably has something to offer as far as handling stress. So you take these seaweed plants and you do extractions. You're trying to find out how do they do this. So there's high temperature and low temperature combined with acids and bases, and you mix and match those, low acids, low bases, and you get different things every time you do this. So you get this extract from the seaweed, that light green blob. You put it on a test plant, which is Arabidopsis. They're small. They've got a small genome, right around 24,000 genes entirely in that whole plant. They don't take up much room. They reproduce a lot, so they're a good plant to have as a test subject. You put the little green blob on there. Your Arabidopsis plant responds to that and makes something. You're like, okay, what does that something do inside a plant? You put it on a gene chip. There's 600,000 of these little holes in this gene chip, and each one has 5 genes or 25 copies of genes in there. Some get turned on, some don't. The ones that get turned on, you go, okay, let's figure out what that gene does, and then you start going, okay, now we need to test it on plants. You take your new extract, you put it on some plants, you subject it to heat, cold, drought, moisture, then you run it through these scanners, and these scanners basically take red, green, blue light, and they go, how big is this plant? It assigns a number to the size of the plant. So you see if your extract is helping the plant do something. The near infrared says how good is the water use in this plant. Ultraviolet says how much chlorophyll is it producing. So you're trying to say, did my extract that I put on there, did it help this plant or not? And the root analysis, that's kind of cool because now you can do non-destructive root analysis. It used to be to do a root analysis, you had to take the plant out of the pot, wash the roots. Now that plant's not going to live anymore. and you would see what the roots were doing. Now you can leave it alive and just keep monitoring, okay, what is this doing to the roots? And the key speakers mentioned about soil health, root health. That's going to be a big focus. It's going to be, what can I do to get better roots in my crops? Then that's great. It's on some lab in Italy. We kind of go, well, please send us some. We're going to try it here, see if it does what you say it is going to do here. So that's what Syngenta is about, is trying stuff in local markets. What does Megafold do? Wound response. Plants are trying to do this themselves. They're trying to fix themselves when they get hurt. They're trying to stay alive when they're hot or cold, and they're making proteins all the time. They're going through photosynthesis all the time, and they're moving nutrients up and down. Megafold has substances in it that goes, hey, you wound response gene, you need some help? I'll help you out. You can now do 62 times what you used to be able to do. Same thing with heat, cold, not 62, it's 5, it's 8. The interesting thing is, those are bad things to happen to you, a wound, heat, or cold. The other 3 at the bottom, they happen every day. So if you're putting Megafold on, it's actually helping the plant be a healthier plant most of the time. So you can go, well, I had a decent year. I don't know if that Megafold even helped me. It's like, well, actually it likely did, because it is helping with day-to-day routine stuff in the plant. The wound stuff, though, is right where a grower should say, I just went through hell because I had a brutal hailstorm. Is there anything I can do? Canola, when it's dinged up by hail, tries to fix itself. It sends out glue, basically, and it forms a callus just so that anything on the north side of the injury doesn't turn brown, dry up, and die. You could probably save those pods if that glue holds. So there's a hailstorm, Saskatchewan, July 14th, a hailstorm, July 4th, 2018. They spray some Megafold on the plot, and they recover about 3.2 bushels an acre of canola versus not spraying Megafold. Now, it's pretty hard as a sales rep to forecast for hail. To go, how much Megafold are you going to sell? Well, I'm going to have this hail event and that hail event. That's pretty difficult. So you want people to kind of go, where can I fit it into my everyday thing? We had a trial in Alberta. This is 2021. Basically, potato grower put it on once. That's a mistake on my part. That's what I suggested. And this is what happened. You got a few more potatoes, but the interesting thing is you get paid by the ones at the top, the light green and the dark green. The more dark green ones you have, the better. Those are your bonus tubers. So you do some 10-foot digs, you sort out the potatoes, and you say, how am I going to get paid on this? And by putting on one application of Megafold, again, a mistake, they made a decent return. Then in 2022, got a whole bunch of Megafold trials planned. Don't bother looking at the table on the left. It's more about the picture on the right. And the grower says, yeah, fine, we have lots of these trials. Then we sign the Invita deal. We drop all those Megafold trials, and we're working on Invita. One grower carries on with his 2 trials. He's out in Alberta. He got 2 wheat fields, Brandon wheat fields, and one doesn't yield very much. Our internal rule, and a lot of people in the industry, their rule is if it's below 2 bushels, either way of 0, it's probably field variability. You have to get over 2 bushels return to figure this is now heading in the right direction. Like, this could be real. One field, not much difference. The 3.9 bushel field, that's worth looking into. I've learned over my career that when you get a big bar, nobody has any questions. When you get a little bar, what the hell happened here? And that's when all the questions start coming up. And that's part of my job, is to go out and ask those questions. What happened here? So, also 2022, there's a grower in Eastern Ontario. If you're on Twitter, try and find Mark Davis. Really cool guy. Keynote speaker said something about farmers doing research. That is Mark Davis. He's a really cool guy. Does good work. He does a lot of work because it's multiple reps. It's not just I split a field. He does strips. And each strip is a combine width wide. He's got a 90-foot sprayer, so 1/2 is this, 1/2 is that. Actually, he does 90 feet. That's the easiest way to do it with the sprayer. And he harvests right down the middle with his combine. Not easy to do. You can sound like, that's a lot of work. Yes, it is. And Mark gets really good answers. So he's using Megafold in wheat. I didn't know he was doing this, but he was talking to a sales rep. He knew we had it. He said, what does it do? It's a stress thing. Okay, fine. he puts it on at his first application timing. That's right around growth stage between 32 and 37, or just before flag leaf, just before the crop starts getting taller. And these are the results from his strips that he's harvesting. He harvests a megafoal strip and then the one behind it, or beside it. Megafoal loses. Bummer. Then megafoal wins the next one. You're thinking, okay, we're back to even here. And then the next one, and it's always compared to the one beside it. He's got lots of strips and lots of checks. So you're not comparing a check over here to a strip way the heck over there, because who knows what the field's like over here. It's nice to be in the same neighborhood. So he's going across the field, and these numbers are getting better. Then Mark does something I thought no grower would ever do. He sprays twice. He sprays at the early stage and then with his fusarium spray. And the numbers just keep getting better. So it's like, wow, this is awesome. Mark's going to try this again in 2023. There are 8 sales districts in Canada with Syngenta. Each sales district is going to see if they get the same results next year. Because I didn't know enough to suggest to people, spray this stuff twice. There are certain crops you should probably spray it 6 times, but those have to be pretty high value crops. And that's ½ a liter an acre. The label we got from Valagro says a liter. And we're like, oh, thanks, nobody's going to do that. Everybody immediately says, I know what this stuff is, and they went down to ½ a liter, and he sprayed it twice. So that's the megafoal story. That's stress mitigation. Now we're into this, how do we get nitrogen from the atmosphere? And the product name is Envita. You're all familiar with the Haber-Bosch process. That's how we get fertilizer from the atmosphere now. Take some methane, and you fix the atmospheric nitrogen. You can't do this in your kitchen or your backyard because you need 100 to 200 atmospheres, and it has to be 400 to 500 degrees Celsius. This is not a kitchen experiment. It takes 8,000 calories to make a kilogram of nitrogen. Then we got biologicals, Envita. It works at room temperature. It gets inside the plant, and basically without the methane part, that's a good thing, you take less energy to create a kilogram of nitrogen. There are differences. It has to be inside the plant. The nitrogen fixed by Envita is to supplement applied nitrogen. Don't think you don't need to buy nitrogen anymore. Eventually, that might be the case, maybe in 10 years, maybe in 5, but not today. Don't be saying, I'm going to save this much money on nitrogen. Just try it first and see how you like it. Envita was discovered back in 1988 in sugarcane. Sugarcane takes nitrogen. Applying nitrogen to sugarcane doesn't work that well because it's hot, it's humid, and a lot of it volatilizes before the sugarcane can get at it. So it's a natural fit that this bacteria found sugarcane, and sugarcane liked it. 10 years later, a company gets formed to say, okay, how do we monetize this? How do we get this product to growers? Because right now it's got to get bigger than sugarcane, otherwise it's not going anywhere. 2019, it gets launched, and that's when the real learning begins. Because after 20 years in R&D, I would do 5 years of trials on a product, and the first year we sold it, 5,000 farmers taught me a lot about that product that I'd worked 5 years for. It's just mass. Growers get out there using this stuff, we're going to learn a whole lot more. So let's compare it to something you're familiar with, rhizobia, inoculants. You've been using them for a long time. They form colonies, they make nodules on the roots. That's how you know it's working. You can dig them up, you can count them, you can see them, they're nice and pink, they're working. Like I said, only on the roots, only on legumes. And they're free living. They can survive in the soil, not a problem. Let's look at Envita. It forms colonies, not little nodules. It gets inside the plant and you can't see it. It gets inside plant cells. As the plant grows, it grows with it. It gets in roots and chloroplasts. Chloroplasts they create photosynthesis, right? So chloroplasts are everywhere in a green plant. So Envita is going everywhere. Legumes and non-legumes, the tens of millions of acres of non-legumes. That could be a fit for Envita. It is an obligate endophyte versus free living. You break this down. Obligate. Think of obligation. Think of must. And then endo means inside, and phyte means plant. Must be inside a plant. Otherwise, it can't live. So how does it engage with plants? First of all, it has to be able to swim. I'll show you how it swims. Then if it's going into the roots, they create hydrolytic enzymes. It basically punches a hole in the root and then gets inside that way. You're thinking, oh great, a hole in my root, that sounds like damage. Well, it makes a biofilm to basically seal up the hole first so that nobody else can come in behind it. And it also makes that biofilm to keep from getting attacked itself. Here's a picture of the root. This is how Exotic thought they were going to sell this at first, and they still might. But there's little Invita bacteria out at the back. They're on the outside of the root. That's a dangerous place to be. Other things are going to come and eat you if you don't get in there quick. They make that enzyme, they get inside. Now they're inside, swimming around inside a root. That's also a dangerous place to be because plants have the equivalent of white corpuscles that are looking for invaders. They're going to try and eat it. So get inside a cell. It gets inside a cell, makes a little case around itself, and then it starts eating sugars within the cell, which is fine. The plant's growing. It has sugars to spare. And then it starts making nitrogen for itself. And if the plant needs nitrogen, it draws from Invita, and the Invita just keeps making more nitrogen. This is what it looks like inside. This is an electron microscope image. That's one single colony of Invita, and these are inside a chloroplast, those dark spots. Now, if you're going to do it foliar, this is the little animal or bacteria that is trying to get inside your plant. It's 0.7 millionths of a meter wide and 2 millionths of a meter long. It swims using 1 to 3 of these long hair-like flagella. Now, in foliar application, Invita finds its way in. It doesn't make a hole. It doesn't puncture the leaf at all. It's trying to get in. The number one way is through the stomata. Those are the doorways. Then there's lenticels, which is usually on stems and on barky plants. And then hydrothodes, those are around the edges of the leaf. There are by far more stomata than anything else. So here's another micrograph. The blue things are chloroplasts. The red things are little colonies of Invita. And that big dark thing, that's a stomata. That's the door. So it's pretty easy to see those things getting inside the door. And that's how they get in there. And here's another living micrograph. All these little guys here have all figured out there's energy on the other side of that door. That's where they want to go. So now we're trying to figure out which is better, roots or leaves. So we do a trial. We inject some Invita solution, same as you would be spraying. And we put it in the roots, and then we put it on the leaves. And then we come back after 2 weeks and go, okay, where is it now? And we can analyze for it. So the first thing you do is you actually put some on some roots and leaves, and you go, can we tell what we're looking for? Yes, great. And then, do we have any contamination in the lab? Check some plants you didn't do. No, it's not jumping around. It's not contaminating everything. Good, let's check and see what happened. This is when you put it on the roots. Lots of it's in the roots. Good, we put it there. And then, did any get up to the plant? Yeah, some. Then we go to when we put it on the leaves, and we go, well, look, it's moving down to the roots, no problem. And there's a lot more in the leaves. So I think we've changed course. And instead of saying, let's put it where the roots are, let's put it on the leaves. As long as the stomates are open, that's a much better place to put it. So it's an obligate endophyte, must be inside the plant. So what's good and what's bad for Invita when you're trying to get it inside the plant? If it's dry, that's bad. They desiccate, they die. If it's freezing, that's bad. They're alive. They're going to freeze and die. Moisture is great. Either when you're planting, if you're going the root way and you're going to plant Invita and put Invita in the furrow, you plant to moisture, which is always a good planting method anyway, because that means your seeds are going to germinate faster. But there has to be moisture. Root tissue. If you plant your crop and it just sits there, it doesn't even germinate for a week, Invita is going to die. So you've got about 48 hours in the soil for Invita to colonize that root. But the root has to be there. And you need open stomates if you're putting it on foliar. So once it's in there, what affects how much it's going to work? There has to be carbon. Well, plants are made of carbon, so that's not a problem once it's inside the plant. Nitrogen availability. If you have lots of nitrogen available, Invita is going to make enough nitrogen to survive by itself and not make any more. If you start running out of nitrogen in your plant, like by the stuff you've put on the ground, then Invita starts making more. So if you've got tons of nitrogen, Invita is not going to make a big difference. Oxygen. This is a bigger thing if you're working and you're putting it in the soil, because if you've just plowed and then cultivated, you've incorporated lots of oxygen in the soil, that actually reduces the activity of Invita. Once it gets inside the plant, that's not an issue. Micronutrients. Molybdenum. The way we know molybdenum is critical to Invita is there's a pocket in Ohio that has very low molybdenum levels, and that pocket does not respond to Invita at all. If you put molybdenum into your fertilizer mix, then things turn right around and Invita starts working. Then you need vanadium, iron, sulfur, manganese. Those are, except for the vanadium maybe, the rest of them are all fairly common, and you know what you're doing. Which brings us to Liebig's law of the minimum. Whatever you're lacking the most, that's what's going to limit how your crop performs. I see the staves on this bucket and I'm like, okay, water, light, and oh hey, there's molybdenum right there. So it's not like it's a weird thing. So, best management practices, I'm going to fly through these pretty quick. If you buy some Invita, the rep should be able to tell you all these things. It's got live organisms. Don't let it bake in the sun. Don't let it freeze. Shake it. Doesn't take long to resuspend it. It comes out really easily. Once you open the jug, all those little live things want to be inside a plant as soon as possible. All product must be applied on the same day the jug is opened. It'll last a season. Don't buy it in December because it's already a year old. That's as long as it lives. You're going to apply it in furrow, for example. Apply it seeding and in-furrow application. Avoid applying Invita in prolonged dry soils. Must be applied direct to seed at planting. Don't put it in if you're doing a 2x2 band. You've seen the size of Invita. A 2x2 band, putting it 2 inches away is somebody saying, I'll give you a ride to work, and they drop you off 20 miles away. So that's a long haul for a little Invita thing. It's got to be right on the seed. Water volume. 9.5 liters per acre, 2.5 gallons per acre of water before adding Invita to the tank. Whales, how you're going to mix it, Invita last. Okay, and if you're mixing Invita with fertilizers, try and get one with a low salt index, and you got 6 hours. Once you've mixed it up, get it planted. Because fertilizer is pretty harsh stuff, really, if you're a living organism. Foliar, 95 mils an acre. A jug will treat 40 acres, one case will treat 160 acres. Water volume 10 gallons or more are recommended. 6 potato growers in Alberta tried this year. 2 of them put it on aerial. They did not go at 10 gallons. They went at 2 and it worked out fine. We are recommending 10 gallons for now for your best chance by ground. Mixing order, last. If you have to put an adjuvant in because whatever pesticide you're mixing it with needs an adjuvant, fine. Put your adjuvant in last just because you don't want a lot of foaming going on. If you're putting it on its own, it helps to put in 1 liter per 1,000 of an adjuvant. It just helps spread it out on the leaf a little bit better. It's got a better chance of finding a stomate and getting inside the plant. It's compatible with most leading pesticides. Mixing is not a problem. One thing to know though is you're mixing it with a phenoxy, the clock is ticking. Because phenoxies have natural bacterial properties. They want to kill bacteria. So you're mixing up 2,4-D, MCPA with whatever grass herbicide you're putting on. You get to the field, throw in your Invita, go. Don't mix it up and then drive 10 kilometers because that bromoxynil or whatever is trying to break down that Invita the whole time you're driving. So throw your Invita in at the last minute. Don't mix Invita with copper. Copper kills bacteria. Invita is a bacteria. Makes sense that you wouldn't want to mix it with copper. Don't put it in with straight 28% or 32% for that matter. You know what it's like when you get that stuff on a hangnail? Imagine you're an Invita, like just crisp right up. So you have to dilute it with water and throw the Invita in last. Spray the solution immediately, especially if you put fertilizer in. It's a numbers game. There are trillions and trillions of Invita inside a jug. And then once you pour them in, they start dying. Your goal is to get half of what you put in there onto the plants alive, and then they'll take care of the rest. Doesn't like to bake in the sun. Avoid spraying above 25. First thing in the morning is ideal. And so if you're going to start mixing it with Liberty, and you're having a great spray day, and it's 3 o'clock in the afternoon, and it's 32 degrees, your Liberty's going to work awesome. Your Invita's going to die. So it's hard to find that period, just say, okay, I'm going to stop throwing the Invita in because now I'm past the time of day that it likes. It's starting to sound like it's a really hard product to use. But think of other things you use where you kind of go, yeah, I know I shouldn't be doing this. I know I shouldn't be doing that. Who goes out and sprays glyphosate in the rain? Not a good idea. Who knows if clouds are coming in to spray glyphosate? No. There's other herbicides you don't spray if you don't think it's going to rain. Other herbicides need rain. So there are all sorts of things. You don't spray glyphosate after a heavy frost because it's not going to kill anything. It's all still asleep. So there are other products that you've gotten used to how to handle the restrictions. Rain fast in 2 hours. That's about how long the maximum time it'll take to get inside a plant. And for now, we're saying get it on early because you want it to grow with the plant so that when your plant hits that nitrogen deficit, Invita is there to fix that deficit. So we did a lot of work this year. At least in my mind, it was a lot of work. And the results are trickling in. I mean trickling. We don't have a quarter of this data yet, but I'm going to share some of it, and some of it doesn't look. There are a lot more questions than there are answers sometimes. This minus 2.7 in a canola field. Who wants that? Not many people. That field gets a lot of hog manure. Didn't need Invita at all. Same growers go to a field that doesn't have hog manure and get this. So, lesson learned. If you have access to livestock manure, you probably don't need to put Invita on that field. And then there's more data to come in. Same thing with wheat. There's lots of good examples on the right. There's one guy I have to talk to. I say guy. There's one grower I have to talk to. They got -7. I need to know what happened there. There are other stories in here. There's a couple of trials in Davison, Saskatchewan. They didn't see a big yield differences, but they did see 0.5% increase in protein. So there might be something there. Other reps are taking notes in these trials and going, this is a 0 because there was a huge patch of wild oats in the middle of my Invita section. And then the other side was fine. So who knows how much those wild oats were costing, but they put the data in the way it was. So we hopefully have a better story next year. This is Ontario, winter wheat. Here's a grower, it's in fine print, don't read it. He's playing with his fertilizer rates to see what can I get back from Invita. And this just reinforces, do what you're doing now, and Invita will help you now. Figure out how much you can cut later. And then the tall green bar, these bars, that's what he's getting over 107 bushels an acre. So it's helping even in a really good yield environment. The higher one that's 9, that grower got 97 bushels, and this trial was actually 9 bushels more. And then there's potatoes. Those are in hundredweight. So 12 hundredweight. The dark green bars, those are irrigated. They're from Alberta. And usually you lose 1 or 2. You've just seen graphs in other crops. There's always some below the line. There are none here. Potatoes. This is something that potato growers should really be considering. And the dark green bars are irrigated, so that doesn't fit the rest of the country because how much of the country can irrigate? Not like they can in Alberta. Ontario, 12 hundredweight. This is done by farmer math. Most of them were done with digs. You dig 10 feet, you do 3 reps in each treatment, and you separate out the tubers. The little green one on the end, that's a farmer going, how far do I have to drive before the truck's full? And he's driving shorter distances for full trucks on the Invita side. Do the math and kind of go, it's about 1,200 weight an acre. Say Invita costs $15, 100 weight of potatoes worth about 10. So spent 15, got 120 back. And he's the lowest in the trials. And that grower had 77% of the 30-year average rainfall. So Invita's helping him out in a dryish year. The other green bars, light green bars, are also not irrigated. 2 in PEI so far. There's another 10 to come from PEI. And they're all heading in the right direction. PEI right now, 100 weight of potatoes is $12. So you can see the return is actually pretty good. And then there's one in Quebec. I'm disappointed in this trial. It's a ridiculous yield result, but they didn't do enough digs to make it reliable. So it's off the chart. They got over 200 weight more, but they only did 1 dig. And my guess is they just figured, this is awesome. Go get the digger and let's start harvesting. So they didn't do the work to make it reliable in my mind. So having seen all that, you just kind of think, where is the fit for biologicals on my farm? And some place to start might be, you've got a field that's always just a little disappointing. Like, man, it looked great, and I thought it was going to yield this, and it just didn't. So what can I do? And biologicals would be a place to start for one of those fields. Okay, and that's it. There's a great big screen of all sorts of cover your butt stuff. Any questions? Go ahead.
I have a couple questions, but so just to clarify, you guys are supplying the Invita to Azotic?
No, it's the other way around.
Or they're supplying it to you and then you guys are marketing?
Correct.
Okay. And then are you doing any tests, like tissue tests or anything like that to determine like what nitrogen levels are in the plants?
Yes, some people are doing PDO testing. We'll be doing a lot more of that next year. Potato growers said that they have their scout do tissue testing every 2 weeks, and their un-Inviticide is doing this, and their Invita, this is for nitrogen, and their Inviticide is just staying flat. So that's how they could tell Invita was working. And they thought that was really cool. What we're working towards is, if a grower says, I sprayed Invita a week ago, how do I know it's in there? We want to get a bioassay test where you take a leaf tissue and then just scrub it into one of those wells, and if it turns green, it's there. If it doesn't turn green, it's not there. But we don't have that test available yet.
Do you have any other products that you're bringing out?
Yes, there's several. Philagra has several, and of all those companies that were on one of the earlier slides, we're always watching what they're doing. And a lot of times they're really good at inventing stuff, and they don't have BSF or Bayer or our critical or Corteva's critical mass to go, how am I going to sell this? Because Azotic is like 5 people in Canada, and we're more. New stuff we have coming out, sometimes it's code numbers, sometimes it's code names. The hard part in my mind is going from lab stuff to small plot stuff. That's okay, it's kind of proof of concept, and then a farmer. And this is where I could really use more Mark Davis's to kind of go, like when I was describing how he does it, I could see some people going, I'm not doing that. And it's like, well, there's probably a little bit of pocket change in there for you. Because side by sides, when you win, it's great. When you lose, not so good. And you're kind of going, well, was it field difference or what's the deal? You do strips, and basically you're taking the field variability, or you're reducing the field variability effect. Then it kind of tells you, okay, this is real. So as a grower, just pencil it out and go, how much of a pain is it for me to do this? Like, what's it worth? When would I say, yeah, okay, I'll do that? And just put a dollar value on it. Just say, tell you what, Syngenta, I'll do a trial for you. It's going to cost $5,000. $250 up front, or $2,500 up front, $2,500 when I give you the data. We're not giving you $5,000 up front. So you think about it. And $5,000 is just my made-up number. You decide what the market will bear.
Yeah, we had some really positive stuff with results with nutrition.
Yes.
Yeah. I haven't seen, we have, again, the trickling in the data.
Yeah.
Don't have much for Invita.
Right.
Yeah, it's
Yeah.
high-yielding fields.
Yep.
Bit of a surprise. It's good.
Yeah. Yes, sir.
So how long do the bacteria last in the plant?
Until the plant starts to dry down.
Okay. So
And
they reproduce, or they just live that long?
they reproduce. They reproduce. In the jug, they don't reproduce. They figured out, this is a 19th version of Invita. And this one, they've figured out, okay, how do we put it to sleep to go in a jug and live for a year and then go nuts when they get out on a plant? And then when they freeze at the end of the year, or when there's no chlorophyll production happening in the chloroplast, they just die. And that's why we're thinking, how's this going to work in permanent crops? You know, is Invita going to survive inside a tree over winter? Don't know. So until we have that tissue.
Did you have it with fungicide? I think the timing was pre-fungicide, right?
Nope. No, the one trial, the one with all the bars, where the guy does strips, he mixed it with his fungicide. Both fungicides, his early one and his later one. So it mixes with fungicides just fine. One of our new things that's coming along, on its own, when you put it in a trial, it does nothing. And you're like, oh, how the hell are we going to sell this? And then you put it with the fungicide, and compared to the fungicide alone, it adds about 25% to what the fungicide can do for you on the yield side. So it's like, okay, here's this biological that doesn't really do anything because it can't control disease. Plant gets disease, it's not doing much. You control that disease, now you're getting something from this biological. So as if you're Syngenta selling a lot of fungicides, you're like, that's the way we want to sell stuff. It's like, throw this in with your fungicide. And luckily, it seemed to work really well. Because in the trials, if you just looked at this product on its own, you'd kind of think this is going nowhere. You have questions, sir?
Yeah, it kind of second part to that question. So if it lives in the plant season long,
Mm-hmm.
we'd want it on early.
Right.
Three usage of canola or
Yep.
herbicide.
Yep.
No need to split out.
Nope.
Put 2 applications on in a season.
Only if you thought, I put it on and it froze or it desiccated. Correct.
Second question is in furrow or seed applied.
Yep.
Will it survive in the soil long enough for roots to germinate and infect? Or what's the time there we have?
2 days, 48 hours.
So we need to get a root out in 48 hours.
That's right. A nub. And you're like, oh, that's never going to work.
Well, it depends when we seed. So in Saskatchewan, we're seeding very early.
Right. I've seen the same thing.
Well, it depends. Sometimes we have good moisture, but we're seeding like Durham at your temperature you had it, right? So
Yeah.
it's taking longer than 48 hours to get it. Typically.
Yeah. My home exam.
In furrow and then foliar?
I don't know. I don't know. After 20 years, I learned that that's a valid answer. I don't know. The other thing, like you talk about crops, like really, how's the root going to be there in 2 days? Soybeans. Soybeans planted on a warm afternoon, they're out of the ground in 5 days. So they've been doing a lot of work below ground before that. So growers have seen, no, I prefer in furrow and soybeans. It's like, well, okay, fine, good for you. Rule of thumb, I'd spray foliar.
It'd be crop dependent, I agree.
Yeah.
We need canola to germinate. It works very quickly.
Yeah. Yeah.
And how much is it?
It's right around $15. I don't like to talk price because I don't think anybody's paying what we suggest they pay. Your retailers are probably giving you a deal as a rule, but that's their business.
And have you been able to measure how much less nitrogen or how much more nitrogen it produces? Or how much less you have to use? Like, is there any other?
Very little. We have very little data. I will tell you a story that you will immediately forget. Okay? Growers put this out in the field. I know you're not going to forget it. They put it out. This is only one example, so that's what it's worth. One example. They split a field. I love when they split a field, rather than these strips. And they sample the Inviticide, and they go, I saw no yield difference whatsoever. They go out and they sample the field. There's 15 pounds more nitrogen on the Inviticide left in the soil than there is on the other side. One person has done this. They're still soil sampling now, and we're asking for more people to send us that data. So we'll see if 15 holds or if it starts dribbling down. And every individual story means more than the average. Like the average kind of just hides a lot of information in there. What's a lot more useful is individual stories. So,
You're looking like more trial sites in 23?
yes. Well, we also have to sell a lot too. That's part of our agreement with Exotic. But I would think that if growers want to share their information, we should make it worth their while to do so. Not the $5,000 mark maybe, but something.
So Syngenta will be doing their own R&D on this along with Exotic.
Exotic's still responsible for doing most of the work. We will suggest what they should be looking at because it's interesting. It's almost like a bunch of lab scientists found this stuff and had no idea how farmers were going to use it. And we asked a whole whack of questions, and they had the same answer I did about in furrow and foliar. I don't know. So we're learning a lot. We've learned some, and I think things are just going to get better with, growers responding to requests to do trials. And some guys can't lose. Potato growers, a lot of them are just going, yes, thank you for that 40 acres. It was awesome. How much are you thinking to buy it next year? Well, I got 500 acres of potatoes next year. How much does it take to do 500 acres? That's dangerous because the question should be, have you got a field, are all your fields like this? No. Well, then let's go through field by field. See this one here, mostly clay? I bet you a well, you don't grow potatoes in clay, but if there's more clay than sand, you would go, this field is probably, you're not going to see the same results in this field. If you want to, go ahead. But I would say, try it on the other ones. This one, probably beyond how Envita is going to help it, or it doesn't need it. Or, now you don't put manure on potatoes either because you tend to get a lot of scab if you do. So growers have learned don't do that. So they might just have residual, like, oh, this was in ground cover last year and I plowed it all down and blah, blah. It's like, there's tons of nitrogen in there. I don't think you're going to see a big, huge bump from Envita.